the enzymes are synthesized. Okay. So at the same time, all enzymes are proteins, all proteins are not enzymes. Okay. So here we also discussed about the specific characters or the physical characters of this catalyst, which is called the enzyme. So they are colloidal in nature, they are thermolabile, they are specific, and they are water soluble. And we also discussed about the biomedical importance of the enzymes. So mainly it is not only in the formation of the other products. The enzymes are not only involved in the formation of the other products, it is also involved in the diagnostic process also, like the liver diseases, like the cardiac failures. At that time also, the enzymes which are present in that organ will be elevated. Okay, so for the diagnostic process also, the enzymes are important. And they are classified into two types. One is called the intracellular and extracellular enzymes. Intracellular enzymes are called the enzymes which are present inside the cell, extracellular are the outside the cell. So here in the extracellular enzymes, the digestive enzymes, all the digestive enzymes will comes under the extracellular enzymes. Okay. So at the same time, we discussed about whenever we are talking about the enzyme, we have to write the suffix ASE, like urease, lipase, glucokinase, hexokinase, like that. How can we identify the enzyme is by seeing the suffix ASE, prefix and suffix. Okay, this is the suffix here. So some exceptional cases are there. What are the exceptional cases there? The trypsin, chymotrypsin, pepsin, all these are the exceptional cases. Okay, only 3-4 are there which are exceptional cases. Remaining all the enzymes are having the enzyme ASE as a suffix. So we also discussed about the zymogens. What are zymogens? They are the inactive forms. Okay, so their inactive forms are called the zymogens, also called as the proenzymes. So if you are talking about trypsinogen, chymotrypsinogen, pepsinogen, all these are the inactive forms. So that means all enzymes are totally not active in certain state. Okay, so whenever the food products are there in our body, at that time the food products will stimulate these enzymes. Okay, they will stimulate the inactive form of the enzyme to convert into an active form of the enzyme. Okay. Again, we classified the classification. How we classified? Based upon the IUB-MB. What is IUB-MB stands for? International Union of Biochemistry and Biology. We classified the enzymes into how many classes? 7 classes. Okay. What are 7 classes? Oxidative, transferase, hydrolase, isomerase, ligase, and ligase. So here, for easy thing, we can write this in this way. So OT means operation theatre at the hilltop. OT means O stands for oxidative, T stands for transferase, H stands for hydrolase, I stands for isomerase, L stands for lyase, and one more L stands for ligase, and the last one is called the transferase. So that's why to make it understand or keep it in your brain, so we are giving the same this one called OTHILLT. That means operation theatre on the hilltop. Okay, normally operation theatre is in the fourth or third floor only. Okay, so that's why we are giving the name that operation theatre on the hilltop. Okay, so OTHILLT. So again, we discussed about the oxidative phase. What are oxidative phase? Means simply when we are talking about, so it will transfer the hydrogen atoms. Simply, okay, to transfer the hydrogen atoms from the one substrate to the other form to form a product. Okay, so the oxidative, we give an example. What is that example? Pyruvate is converting into lactate. Pyruvate is converting into lactate, where the hydrogen molecules are transferred from one place to the other. That means the hydrogen molecules are attached to the pyruvate, so that it's converting into a lactate form. Okay. And the second one is called the transferase. So what is transferase means? Not simply transferring of hydrogen atoms is by the help of the oxidoreductase enzyme. Transfer of hydrogen atoms by the oxidoreductase enzyme. So the other things which are transferred, like the amino group or the carboxyl group or the methyl group, they are transferred by the help of an enzyme called transferases. Okay, oxidators will transfer only the H plus ions. Will transfer only the H plus ions, and the remaining are be transferred by the help of the enzyme called transferases. Okay, so we have given some examples here also. That what are the examples? Aminotransferase, okay, alanine transferase, transcarboxylase, transmethylase, and kinases. Okay, so in that we have given the example one is called the aminotransferase. Means what will transfer? What will transfer? Transfer the amino group. Okay, so second one is the transcarboxylase. What will transfer? Transfer the carboxyl group. And third one is the transmethylase. What will transfer?
Methyl group.
Transfer the methyl group. And last one is the kinases. Means here it will transfer the phosphate group. So here I gave an example of kinase. Okay, where the glucose converting into glucose 6-phosphate. You can see the structure there. The glucose is having a 6 carbon sugar. So as a 6th carbon sugar, then 6 carbon compound, isn't it? So it's a 6th carbon atom. It's a 6th carbon atom. It's the 6th carbon atom. To that, the phosphate group is attached. From where the phosphate group is obtaining from? Where the phosphate group is obtaining from? From the ATP. That means the ATP, adenosine triphosphate, is donating its phosphate group to the glucose molecule, which is going and binding to the 6th carbon of the glucose molecule, so that the glucose converting into glucose 6-phosphate. Okay, so for that what the enzyme required? Hexokinase or glucokinase. So what are the kinases? Whenever you type simply, wherever you are getting the ATP, you can write kinase. Okay. We also discussed about the next one is called the hydrolase. What's hydrolase? Lyase means breaking down and in presence of water molecule. Hydrolases. Okay. So in presence of water molecule, the substrate molecule is converting into two structures. So that's called the, by the help of the enzyme, hydrolase. That means whatever the digestive enzymes are there, all the digestive enzymes are broken down by the help of water molecule only. Like sucrase, lactase, maltase, all these enzymes are there which will break up only with the help of the water molecule. Okay, so that's why we are getting it an hydrolase. So I have given the example also here. The lactose is the milk sugar. Lactose is the milk sugar. So this lactose converting into two monosaccharide units. What are the two monosaccharide units there? Glucose and galactose. Glucose and galactose. So by the help of a water molecule, the lactose, by the help of the enzyme lactase, in presence of water molecule, it is converting into glucose and galactose. Okay, so this is about the hydrolase. And next one is the lyase. What is lyase means? Lyase means in the absence of oxygen. In the absence of water molecule. In the absence of water molecule, the substrate will be broken down. Okay. So what are the breakdown products are the aldolase fumarase here. I have given an example called the aldolase, which is seen in the glycolysis step only. Okay, where the glucose converting into glucose 6-phosphate, glucose 6-phosphate is converted into fructose 6-phosphate, fructose 6-phosphate converting into fructose 1,6-bisphosphate. This fructose 1,6-bisphosphate is a 6-carbon sugar. So this 6-carbon sugar is broken down into two 3-carbon compounds. What are those? One is called the glyceraldehyde and second one is called the dihydroxyacetone phosphate, also simply called DHAP. They are called as DHAP. So without any water molecule, that 6-carbon sugar is converting into two 3-carbon molecules. Okay. So glyceraldehyde is also a 3-carbon and dihydroxyphosphate is also a 3-carbon molecule. Okay. These are the lyases. Coming to the next one called isomerase. So here we are talking about the isomerization. What is isomerization? Simply the rearrangement of atoms. The rearrangement of atoms. But we are not adding or we are not deleting any of the atom. Just we are changing the shape. That means the hydrogen atoms are shifting from one position to the other position of the same carbon atom, different carbon atoms. Okay. So this is called isomerization. So one more time. So isomerization means so we are not adding or we are not deleting anything. But what we are doing, we are simply changing the hydrogen atoms from one carbon to the other carbon atom by the help of the isomerization enzyme. Okay, the enzymes are called the isomerases. Okay, so here I have given some examples like epimerase, isomerase, and mutase and resmase. Just you people can write simply one or two, and that will be enough for people. Okay. So here I have given an example of glyceraldehyde. Glyceraldehyde, see, in the glycolysis step also. In the glycolysis, we are talking glycolysis, isn't it? So glucose converting in glucose 6-phosphate, then to fructose 6-phosphate, then to fructose 1,6-bisphosphate. The fructose 1,6-bisphosphate is breaking up into two 3-carbon compounds. What are those? One is the glyceraldehyde 3-phosphate, the second one is the dihydroxyester phosphate. So what is the end product of glycolysis? Pyruvate. So if one glucose molecule is broken down, how many pyruvate molecules are formed up there? Two pyruvate molecules. How can that be two pyruvate molecules? That means the glyceraldehyde 3-phosphate is converting into pyruvate. Glyceraldehyde 3-phosphate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoryl pyruvate, and pyruvate, isn't it? Glyceraldehyde 3-phosphate, fructose 1,6-bisphosphate is breaking up into two carbons. One is the glyceraldehyde 3-phosphate and dihydroxyester phosphate. This glyceraldehyde 3-phosphate is converting into 3-phosphoglycerate, 2-phosphoglycerate, phosphoryl pyruvate, and pyruvate. So this one pyruvate is obtained, we are getting it. Okay, what is the other pyruvate where we are getting? From the dihydroxyester phosphate. From the dihydroxyester phosphate, again it can be converted back into glyceraldehyde 3-phosphate. That is called isomerization reaction. Okay. So the glyceraldehyde 3-phosphate is converting into dihydroxyester phosphate, or else the dihydroxyester phosphate can also be converted into glyceraldehyde 3-phosphate. They are interconvertible. These two are interconvertible. I have given single arrow, but you can keep both arrows there. So it's an interconvertible. Okay. So glyceraldehyde 3-phosphate is converting into pyruvate. At the same time, the dihydroxyester phosphate will also convert into glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate is converted to 3-phosphoglycerate, 2-phosphoglycerate, phosphoryl pyruvate, and pyruvate. So that's why whenever we're talking about glycolysis, the glycolysis is giving the two pyruvate products. Next one is the ligase. Ligase means ligation, means addition. So if you are adding two small molecules so that it will convert into a bigger one by the help of the enzyme called ligase. So that's called ligation. Okay. So ligating two simple compounds and make it into a bigger one with the simultaneous hydrolysis of the ATP. So here the ATP is hydrolyzed there. So here I have given few examples. One is called pyruvate carboxylase, acetyl-CoA carboxylase, and succinate thiokinase. Okay. So you can see here, the example is the pyruvate carboxylase. That means you can see the pyruvate is just one more substrate molecule. So it is simple one, and one more small structure is the carbon dioxide. Both the pyruvate and carbon dioxide are combined each other to form a structure called oxaloacetate by the help of pyruvate carboxylase. So here the carboxylation step is taking place here. Okay. So that means what is ligation? Ligating two simple compounds and making into a bigger one, isn't it? So here there are two simple compounds. One is the pyruvate and second one is the carbon dioxide. These two simple compounds are combined each other by the help of the enzyme pyruvate carboxylase, forming a bigger molecule called the oxaloacetate. Have you heard this word oxaloacetate? Yes. In the Krebs cycle we are having this. Okay. Next, the last one is called the translocase. Translocase. So this translocase they recently identified in 2018 only in the August. Okay. So up to now we are only having 6 enzymes, different classes, the 6 classes of enzymes. So the 7th class enzyme is recently updated. That is called the August 2018 only they are updated this one. So that enzyme is called the translocase. Okay. So it is the movement of molecules or ions across the cell membrane. So by this we have completed the classification of the enzymes. Okay. So how many classes of enzymes are there? So 7 classes. What are those? One is by simply OTHILLT. One is called the oxidorreductase. Second one is called the transferase. H stands for the hydrolase. I stands for the isomerase. L stands for the ligase. And one more L stands for the ligase. And the last T stands for the transferase. Okay. So what oxidorreductase will do? Oxidorreductase will transfer the hydrogen atoms. Simply for the oxidation reduction reactions it will take place. Okay. So and what is the next one? That's called the transferase. What will transfer? It transfers the amino group, it transfers the carboxyl group, or it transfers the phosphate group, or it transfers the methyl group also. Isn't it? So basing upon that we will give the names. Okay. I have given an example of kinases where the glucokinase and hexokinase will be there. And that's transferase. And next one is called the hydrolase. What is the example of hydrolase? What hydrolase means? In presence of water molecule, the substrate molecules are breaking up into 2 carbon compound, 2 monosaccharide units. Like in the digestion process, we are having the lactose or the maltose. The lactose converting into glucose and galactose by the help of this enzyme called the lactase in presence of water molecule. Okay. So that is hydrolase enzyme. And the next one is called the lyase. What is lyase? So okay, hydrolase, after that isomerase. Okay, what isomerase? It will isomerase. What isomerase? That means the same 3 carbon compound will be same like that only, but no addition and no deletion of the atoms there. Okay, so that is example we had. What example we had given that? This are 3 phosphate into dihydroxyester phosphate. So DHAP and dihydroxyester phosphate is converted into this 3 phosphate. That means it's called a isomerization. Okay, and next one is lyase. What lyase means? Without that means in the absence of water molecule, the substrate is broken down into 2 compounds. What are those? Can you give an example? Aldolase. What the aldolase will do? Fructose once this phosphate is converted into this called 3-phosphate and dihydroxy acetone phosphate. Okay, so that is about the ligase. And the last one is the ligase. One more is called the L stands for the ligase. What is ligase stands for? It ligation of two simple units and combined to form a bigger molecule. So I have given an example. Pyruvate is binding with carbon dioxide molecule to form a pyruvate carboxylase. By the help of pyruvate carboxylase, by the help of enzyme pyruvate carboxylase, we are getting one such product called the oxalo estate. So this oxalo estate is involved in the TCA cycle. Okay. The last one is called the translocase. So what will do? The translocase will assist the movement of molecules or ions. Okay. Come to the next one, is called the holo enzyme. So, first of all, అయితే enzymes are catalyst. They will catalyze the reaction. Okay. So, along with the enzymes, it should require some other cofactors. What are the cofactors? Like some vitamins, which are called the B complex vitamins, or some minerals. మనం తీసుకున్న ఫుడ్లో బి కాంప్లెక్స్ వైటమిన్స్ తీసుకుంటున్నాము. మనకు ఫీవర్ ఎక్కువగా వచ్చింది అనుకోండి, వాడు నీకు ఎలాగో ఎనర్జీ కావాలని చెప్పేసి ఎక్స్ట్రాగా బి కాంప్లెక్స్ టాబ్లెట్స్ ఇస్తా ఉన్నాడు. సో వాట్ బి కాంప్లెక్స్ టాబ్లెట్స్ విల్ డూ? ఈ ఎంజైమ్స్ని యాక్టివేట్ చేయడానికి ఆ బి కాంప్లెక్స్ వైటమిన్స్ కావాలి. అట్ ద సేమ్ టైమ్ సమ్ మినరల్స్. ఓకే, సో దీస్ ఆర్ యాక్టింగ్ యాజ్ అ కోఫాక్టర్స్ దర్. ఓకే, సో normally the single enzyme cannot properly perform, and all the enzymes are not, I'm not talking about all the enzymes, but some enzymes are there, they cannot perform directly. They require some cofactors. Okay, so you can see here, it's an apo enzyme. What is holo enzyme means? What is apo enzyme means? The apo enzyme is the enzyme which is in the inactive form. Okay, so for that one cofactor or coenzyme is required. Vitamin B complex vitamins na vitamins ఉందా తెలుసా? వాట్ ఆర్ వైటమిన్స్? వైటమిన్స్ లో ఫ్యాట్ సాల్యుబుల్ వైటమిన్స్, వాటర్ సాల్యుబుల్ వైటమిన్స్ చదివామా? ఓకే, వాటర్ సాల్యుబుల్ వైటమిన్స్ లో ఏంటి ఉన్నాయి? బి కాంప్లెక్స్ వైటమిన్స్. బి కాంప్లెక్స్ వైటమిన్స్ నేమ్స్ చెప్తారా? థైమిన్, రిబోఫ్లావిన్, నియాసిన్, పిరిడాక్సిన్, ప్యాంథోనిక్ యాసిడ్, ఫోలిక్ యాసిడ్, బయోటిన్స్, ఫైనకోబాల్మిన్. సో ఈ బి కాంప్లెక్స్ వైటమిన్స్ ఏవైతే ఉన్నాయో, దీస్ బి కాంప్లెక్స్ వైటమిన్స్ ఆర్ యాక్టింగ్ యాజ్ ఎ కోఎంజైమ్స్. Directly not active, but what's happening, it will convert into coenzyme form. Like thymine is converted into thymine pyrophosphate. Thymine is converted into thymine pyrophosphate. Riboflavin and riboflavin is converted into FMN and FAD. Okay, so niacin is converted into NAD and NADP. Okay, so like that the B complex vitamins are converting into a coenzymatic forms. At the same time, some other cofactors. What are the other cofactors means? Like magnesium. మనం చెప్తాం గ్లూకోజ్ కన్వర్టింగ్ గ్లూకోజ్ 6-phosphate హెక్సోకైనేస్ ఎంజైమ్ కావాలి, పక్కన ఎప్పుడు మెగ్నీషియం రాస్తూ ఉంటాడు. అవి చూసారు కదా? The magnesium is important. So the magnesium is also acting as a cofactor. That means that is a mineral. The B complex vitamins and these minerals, okay, all are required. So that the enzyme, whenever it's in inactive state, it have to convert it in active state, so that it can bind with a substrate molecule. Okay, so the coenzymes are the enzymes means the B complex vitamins. Okay, and the cofactor, the non-protein part of the factor, so which will both this apo enzyme means the inactive form and this coenzyme will bind with each other, so that it forms a structure called the holo enzyme. Okay, so this holo enzyme is active right now. Okay, enzymes are totally in the inactive state. So I am not talking about all the enzymes. Many of the enzymes are in the inactive state. So this inactive state of the enzymes have to combine with cofactors of the coenzymes. So whenever binding with cofactors of coenzymes, it will convert into a form called holo enzyme. So this holo enzyme will bind with the substrate. Substrate అంటే ఏంటి అక్కడ? ఇప్పుడు గ్లూకోజ్ కన్వర్ట్ ఇంటూ గ్లూకోజ్ 6-phosphate అని చెప్తున్నాము. సో ఎంజైమ్ చెప్తున్నాము గ్లూకోకైనేస్ కానీ హెక్సోకైనేస్ కానీ ఎంజైమ్ చెప్తున్నాం, అవునా? సో గ్లూకోకైనేస్ హెక్సోకైనేస్ ఇవి ఎంజైమ్స్. Glucose, pine structure, glucose substrate molecule. Product ఏంటి? Glucose 6-phosphate. Okay, so that means the glucose in the glycolysis step, the glucose is converting into glucose 6-phosphate. That means the glucose which is seen in the upper side, so it's totally a substrate. So on the substrate, the enzyme hexokinase or glucokinase will act. ఆ సబ్స్ట్రేట్ మీద ఈ హెక్సోకైనేస్ కానీ గ్లూకోకైనేస్ కానీ యాక్ట్ చేసి విల్ గెట్ ఏ ప్రొడక్ట్. దట్ ప్రొడక్ట్, ఓకే, దట్ ప్రొడక్ట్ ఈస్ ఎన్ గ్లూకోజ్ 6-phosphate. అయితే ఈ గ్లూకోకైనేస్ హెక్సోకైనేస్ సో ఈ హెక్సోకైనేస్ గ్లూకోకైనేస్ ఎంజైమ్స్, దానికి కొన్ని కోఫాక్టర్ కావాలి. ఏంటది? మెగ్నీషియం. మెగ్నీషియం ఇస్ అన్ యాక్టింగ్ యాజ్ అన్ కోఫాక్టర్ దేర్. ఓకే, that is about the holo enzyme. So whenever the apo enzyme is binding with any coenzyme molecule, so that then only it forms into an active state. It's not in the inactive state. It's converting into an active state. So whenever it's converting into the active state, then only it can bind with the substrate molecule. ఇప్పుడు మనం చెప్తున్నాం సింపుల్గా ఈ గ్లూకోకైనేస్ అనుకోండి. So this is the enzyme called glucokinase or hexokinase. So it requires one coenzyme or cofactor. Okay, so the cofactor is magnesium. Okay, whenever it's binding, it's forming a holo enzyme so that it will bind with substrate molecule. What substrate molecule there? The glucose is the substrate molecule. So it will bind with the glucose as substrate molecule to form a product called a product form of glucose 6-phosphate of form. Okay, that's about the holo enzyme. Simple write holo enzyme is equal to apo enzyme plus cofactor. Pine red color prime. That would be holo enzyme is equal to apo enzyme, which is the inactive form. In fact, last one is the inactive form plus cofactor. So here the cofactor enzymes, all enzymes are proteins, but the cofactors are not enzymes. అది బి కాంప్లెక్స్ వైటమిన్ అవని, మెగ్నీషియం మినరల్ అవని, అదంతా ప్రోటీన్స్ కాదు కదా. సో దట్ మీన్స్ ఇట్ రిక్వైర్స్ వన్ అదర్ నాన్ ప్రోటీన్ స్ట్రక్చర్ సో దట్ ఇట్ కన్వర్ట్ ఇంటు ఎన్ హోలో ఎంజైమ్ దేర్. కోఫాక్టర్స్ So what are the different cofactors? One is the metal cofactor and some organic cofactors are there. The metal cofactors are metal enzymes, simply called the metal enzymes, and some metal activated enzymes. Coming to the organic factors, the coenzymes and the prosthetic group. So మనం డైరెక్ట్ గా ఎగ్జాంపుల్ గా ఇది ఇచ్చాము. ఓకే, metal enzymes and coenzymes. Okay. సో కోఎంజైమ్స్, అందులో కోఎంజైమ్స్ అంటే ఏంటి మనం చెప్పాము. ఏంటిది? బి కాంప్లెక్స్ వైటమిన్స్ B1, B2, B3 రాసింది అక్కడ. సో B1 స్టాండ్స్ ఫర్ ద థైమిన్. ఓకే, ద active form is the thymine pyrophosphate. TPP is called the thymine pyrophosphate. Like the B2, the vitamin B complex vitamin B2 is called the vitamin B complex vitamin. They called the riboflavin, isn't it? So this active form or the coenzyme form is FMN and FAD. And third one is the B3 niacin. So the active forms are NAD and NADP. Like the B6, which is also called pyridoxal, the active form is PLP, pyridoxal phosphate. So the coenzymes are the non-protein part organic molecules required by the enzyme for their optimum activity. So what I have written, the coenzymes are the non-protein part of the organic molecule required by the enzyme for their optimum activity. So most of the coenzymes are derivatives of the vitamin B complex. చదువుతూ రాయండి, అర్థం అవుతుంది మీకు. ఓకే, మీరేం రాస్తున్నా తెలుస్తూ ఉంటుంది మీ ఎగ్జామ్. ఎందుకంటే బిఎంఎస్ వాళ్ళకి చదివే టైం ఉండదు. ఫస్ట్ ఇయర్ ఉంటారేమో కానీ సెకండ్ ఇయర్ మీకు అక్కడ పంపించేస్తారు, మీ యొక్క డిపార్ట్మెంట్స్కి పంపించేస్తారు. సో అక్కడ చదివే టైం కూడా ఉండదు. సో ఎక్కడైతే చెప్తున్నామో అక్కడే వినండి, అక్కడే అర్థం చేసుకోండి, చదవండి, తెలిసిపోతుంది. ఎగ్జామ్ మీకు ఈజీగా ఎంటర్ చేసుకోవచ్చు. ఓకే, సో వాళ్ళ టెక్స్ట్ బుక్స్ యొక్క పేపర్ టెక్స్ట్ బుక్స్ అని చెప్పారా? సత్యనారాయణ చెప్పారా? ఇంకా ఆశాలక ఎవరు చెప్పారు? ఇది సీనియర్స్ చెప్పారా?
బయోకెమిస్ట్రీకి ఏం చెప్పలేదు, బయోకెమిస్ట్రీకి ఏం చెప్పలేదు సార్, ఫిజియాలజీకి చెప్పారు సార్.
సత్యనారాయణ చిన్న బుక్ ఉంటుంది. ఓకే, ఆర్ఎల్స్ ఒకసారి జాస్న మేడం కానీ వచ్చారు హెచ్ఓడి, ఆవిడ క్లాస్ చెప్పేటప్పుడు ఆవిడని అడగండి ఏం బుక్ ప్రిపేర్ చేయాలనేసి ఆవిడ చెప్పినట్టు. నా వరకు అయితే మేడం మన నోట్స్ ఇస్తుందని నాకు తెలీదు. సో నేనైతే నోట్స్ లెక్క ఇస్తాను, జస్ట్ ఆ నోట్స్ చదివిస్తే సరిపోతుంది. దట్ విల్ బి ఎనఫ్ ఫర్ ఇన్ ద ఎగ్జామినేషన్ పర్పస్. ఓకే,
సత్యనారాయణ బాబు.
సో కమింగ్ టు ద కోఫాక్టర్స్, మనం కోఎంజైమ్స్ ని చదివాము. కోఎంజైమ్స్ ఆర్ ద బి కాంప్లెక్స్ వైటమిన్స్. ఓకే, సో కమింగ్ టు ద కోఫాక్టర్స్ మనం చెప్పాం కదా, దట్ మీన్స్ సమ్ అదర్ మినరల్స్ దే ఆర్ యాక్టింగ్ ఇన్ సమ్ కోఫాక్టర్స్. లైక్ వాట్ ఆర్ ద మినరల్స్ దేర్? ఐరన్, జింక్, మెగ్నీషియం, ఆల్ దీస్ ఆర్ ద మినరల్స్. They are also involved in the active formation of the enzyme. Okay, so whenever the enzymes are in the inactive state, which is called the apoenzyme, it have to bind with an coenzyme or the cofactor or both. Okay, so here I discussed about the coenzyme. That coenzymes are the B complex vitamins. Okay, some cofactors are there. The cofactors are the minerals there. Okay, so here I gave some examples. One is called the zinc, magnesium and all. So I discussed about the magnesium also. Okay, some enzyme requires an additional non-protein component for its activity, optimum activity. So that is called the cofactors. So here I give an example of hexokinase, which is the enzyme which is totally in the inactive state. So it have to convert into an active state by the help of the magnesium there. So the magnesium will play an active role to convert the inactive form of the enzyme into the active form. So not only the hexokinase, even the pyruvate kinase also requires the same mineral called the magnesium. And some other enzymes are there like cytochrome, zinc. Okay, those also require that minerals. సింపుల్గా టిక్ పెట్టుకోండి, దీనికి పెట్టుకోండి, టిక్ పెట్టుకోండి, ఇది రాస్తే అయిపోతుంది ఎగ్జాంపుల్గా మెగ్నీషియం గురించి. ఓకే, so the same హియర్ ఐ గివ్ ద ఎగ్జాంపుల్. ఐ టోల్డ్ యూ మనం ఎప్పుడు అంటే కంటిన్యూస్ దీని గురించి ఎక్కువ చెప్తున్నామంటే సింపుల్ మీకు ఈజీగా గుర్తుంటుంది కాబట్టి. సో గ్లూకోజ్ కన్వర్టింగ్ టు glucose 6-phosphate. I told you the ATP is donating its phosphate group. So one of the ATP and any phosphate molecules, 3 phosphate molecules. In the one of the phosphate molecule is going and binding to the glucose carbon to form a structure called the glucose 6-phosphate, and the remaining 2 molecules are converting into ADP and PI. Okay. So for that one metal is required. What is that metal? That's called the magnesium. So it is acting as a cofactor. So these are called metal activated enzymes. In the metal man cheptum cofactors lo kuda no. So whenever talking about some metals, so some are loosely bound, some are tightly bounded. Okay. So these metals are loosely bounded are called metal activated enzymes. ఎప్పుడైతే అవి లూజ్ అంటే ఆ మెగ్నీషియం ఏం చేస్తుందంటే ఎప్పుడైతే ఆ గ్లూకోకైనేస్ కానీ హెక్సోకైనేస్ కానీ ఆ మెటల్ కావాలో ఆ మెటల్ బైండ్ అవుతుంది, హెక్సోకైనేస్ ఎంజైమ్ బైండ్ అవుతుంది. సో దాన్ని కన్వర్ట్ టు ఏ హోలో ఎంజైమ్. సో ఎప్పుడైతే ఆ ఎంజైమ్ తన యొక్క పని కంప్లీట్ చేసుకుంటుందో ద మెగ్నీషియం విల్ డిటాచ్ ఫ్రమ్ దట్. అర్థమైందా? ఇంకోసారి చెప్తాను. హెక్సోకైనేస్ గ్లూకోకైనేస్కి మనం ఏ మెటల్ కావాలన్నా మెగ్నీషియం కావాలన్నా. సో ఎప్పుడైతే ఆ గ్లూకోజ్ మాలిక్యూల్స్ మన బాడీలో ఎక్కువగా కనపడ్డాయో ఆ గ్లూకోకైనేస్ హెక్సోకైనేస్ యాక్టివేట్ అవ్వాలి. అవునా? సో ద ఇన్యాక్టివ్ స్టేట్ని యాక్టివ్ స్టేట్లో కన్వర్ట్ చేయాలంటే ఏం కావాలి? మనకి మెగ్నీషియం కావాలి. ఈ మెగ్నీషియము ఆ హెక్సోకైనేస్ కానీ గ్లూకోకైనేస్కి వెళ్ళి బైండ్ అవుతుంది. బైండ్ అయ్యి ఆ బైండ్ అయిన తర్వాత అది హోలో ఎంజైమ్ ఫామ్ అవుతుంది. హెక్సోకైనేస్ కానీ గ్లూకోకైనేస్ కానీ హోలో ఎంజైమ్గా ఫామ్ అవుతుంది. ఆ హోలో ఎంజైమ్ గ్లూకోజ్ అనే సబ్స్ట్రేట్ మాలిక్యూల్ని ప్రోడక్ట్గా గ్లూకోజ్ 6-phosphate ప్రోడక్ట్గా కన్వర్ట్ చేస్తుంది. ఓకే. ఎప్పుడైతే గ్లూకోజ్ 6-phosphate ప్రోడక్ట్గా కన్వర్ట్ చేసిందో ఆ హెక్సోకైనేస్ నుండి ఆ మెగ్నీషియం మళ్ళీ డిటాచ్ అయిపోతుంది. మళ్ళీ డిటాచ్ అయిపోయి ఇంకో ఎంజైమ్ అంటే ఇంకో హెక్సోకైనేస్కి వెళ్ళి బైండ్ అవుతుంది అన్నమాట. ఓకే. సో దట్స్ వై దీస్ మెటల్స్ ఆర్ టోటలీ లూజ్లీ బౌండెడ్ అండ్ కెన్ బి సెపరేటెడ్ ఈజీలీ. ఓకే. నెక్స్ట్ in the same thing, one more is called metallo enzyme. Metallo enzyme is tightly bonded. Okay. Metal activated enzymes are called the loosely bounded enzymes. Okay. Whereas metallo enzymes are the tightly bonded enzymes. So here I've given some example like iron, copper. So this cannot be separated. So ఇప్పుడు మనం ఏం చెప్పుకున్నాము? హోలో ఎంజైమ్. హోలో ఎంజైమ్ మీన్స్ కంటైన్స్ అపో ఎంజైమ్ and which is bonded to the cofactor or the coenzyme so that it convert into a holo enzyme. Okay. దీని రాయొద్దు, చెప్తాను. ఫస్ట్ ఆఫ్ ఆల్ రండి, మనం చెప్తున్నాము ఎంజైము సబ్స్ట్రేట్. ఎంజైమ్ ఏం చెప్పాము? ఒక ఎగ్జాంపుల్గా ఏం చెప్పాము? ఎంజైమ్ని గ్లూకోకైనేస్ అనేది ఎగ్జాంపుల్గా ఎంజైమ్ చెప్పాము. గ్లూకోకైనేస్ ఆర్ హెక్సోకైనేస్. సబ్స్ట్రేట్ని ఏం చెప్పాము? సబ్స్ట్రేట్ గ్లూకోజ్ ద సబ్స్ట్రేట్. ప్రొడక్ట్ ఏంటి? గ్లూకోజ్ 6-phosphate ప్రొడక్ట్. ఓకే. సో అది గుర్తుంది కదా. సో హౌ ద ఎంజైమ్ విల్ యాక్టివేట్ and after that what the enzyme మనం చెప్పాము ఎంజైమ్ యాక్టివేట్ ఎలా చేస్తున్నాము? either by the help of the cofactor or by the help of the coenzyme forming a holo enzyme there. సో ఇప్పుడు మనం ఏం చెప్తామంటే how the mechanism of enzyme action taking place. ఓకే. ఎలా జరుగుతుందంటే, సో ఎంజైమ్ will bind with the substrate molecule That means the glucokinase or hexokinase will bind with the substrate molecule like glucose to form a structure called enzyme substrate complex. See here. The enzyme is binding with a substrate molecule. So whenever the enzyme is binding with substrate molecule, it forms a structure called the enzyme substrate complex. Up to here, it's totally reversible reactions. The enzyme plus substrate can combine to form an enzyme substrate complex. At the same time, the enzyme substrate complex can be converted back into enzyme and substrate. But after when the enzyme substrate complex is there, this enzyme substrate complex will convert the substrate molecule into a product. What is the product we are told? The glucose 6-phosphate. Okay, and the enzyme will be released out. If a enzyme panenti, myself is an enzyme. I want to convert you the substrate molecules into a product. అయిపోయిన తర్వాత పని ఏంటి మళ్ళీ ఫస్ట్ బ్యాచ్ ఎవరు వస్తే వాళ్ళకి బైండ్ అప్ అవ్వాలి అవునా సో దట్ మీన్స్ ద ఎంజైమ్ ఈస్ బైండింగ్ విత్ సబ్స్ట్రేట్ మాలిక్యూల్ టు ఫార్మ్ ఎ స్ట్రక్చర్ కాల్డ్ ఎంజైమ్ సబ్స్ట్రేట్ కాంప్లెక్స్ జరుగుతుందంట ఎంజైమ్ సబ్స్ట్రేట్ కాంప్లెక్స్ జరుగుతుంది ఓకే ద ఫస్ట్ ఇయర్ విల్ బి టోటలీ ఎంజైమ్ సబ్స్ట్రేట్ కాంప్లెక్స్ ఓన్లీ ఓకే సో after the enzyme substrate complex you will write the examination and you will convert it into a product. Okay, so what I will do again, the enzyme will be detached from the substrate molecule and I will bind with the other new substrate molecules. Okay, so the substrate is bound through multiple non-covalent interactions at the active site of the enzyme, forming an enzyme substrate complex subsequently converted to product and free enzyme. Okay, so what are interactions taking place there? Non-covalent interactions. What type of interactions are there between the enzyme and substrate? The enzyme substrate binding is called the non-covalent interactions. Okay, so ikkada a enzyme ki ekkada padithe akkadiki bind ayipodam avadu. Only that means active site antaru. Idi enzyme anukondi. Okay, enzyme ki chuttupakkala ekkada padithe akkada bind ayipothe substrate molecule adi product ga form avadu. Okay, enzyme is having one site. That site is called the active site. To that active site only the substrate molecule have to bind and so that it can convert into a product. Okay, ఇది enzyme. Enzyme ki ikkadiki bind aina substrate molecule adi product form avudandi. So when it convert into product only if the substrate molecule will go and bind to the active site of the enzyme. So then only the active site of the enzyme is that the substrate molecule is binding up there so that the substrate molecule is converted into product there. Okay, so that's what we are taking here. Substrate is bound to multiple non-covalent interactions at the active site of the enzyme, forming an enzyme